Agriculture has profoundly changed in recent decades. Large-scale monocultures, the use of pesticides and fertilizers, and a small number of high-yield varieties now ensure food security for a growing world population. But this development has its consequences. Many old crop varieties are disappearing, and with them, a significant part of our genetic heritage. To address this development, the first gene banks were established at the beginning of the 20th century.
Genbanken sind lebendige Archive genetischer Ressourcen, also alten Sorten, die heute nicht mehr im Anbau sind, oder auch wildverwandte Arten heutiger Kulturpflanzen, deren Merkmale aber für die Züchtung von Bedeutung sein können und deswegen für die Nachwelt, für kommende Generationen erhalten werden müssen.
One of the largest and most important gene banks in the world is located today in Gattersleben at the Leibniz-Institute of Plant Genetics and Crop Plant Research. Together with the sub-collections in Mecklenburg, Western Pomerania, more than 150,000 samples, so-called accessions, are preserved. They originate from over 3,000 species, ranging from cereals and legumes to vegetables, herbs, and forage grasses. The collection is preserved, evaluated, and made available nationally and internationally for research, associations, private individuals, and breeders. Most of the collection at the Gene Bank in Gattersleben is stored as seeds in large cold rooms at -18 degrees Celsius. Under these conditions, they remain viable for decades. However, if the germination capacity declines or the stock becomes scarce, new plants are cultivated to obtain fresh seeds. Depending on the type of crop, this is done in the open field or in greenhouses. This reproductive cultivation requires the most careful attention. The different accessions are not allowed to mix or pollinate each other. This preserves genetic purity. Crop species that cannot be preserved via seeds are propagated vegetatively in permanent gardens or in vitro in test tubes. In this process, shoot segments are placed on a culture medium and grown in climate-controlled chambers. And in so-called cryopreservation, dividing tissue is frozen in liquid nitrogen at -196 degrees Celsius. This keeps it viable for at least 100 years. The more information available about an accession, the better a researcher or breeder can judge whether they can use it for their purposes. The focus of the research is on yield, ingredients, but also on resistance to pathogens and insect pests. And, in view of climate change, also characteristics that lead to greater tolerance to heat and drought. The genes responsible for this are anchored in the genome of the plants. Modern sequencing technologies make it possible to examine hundreds of samples per day. But the genetic diversity and family relationships of the accessions can also be made visible. The Genebank's Documentation Centre processes all data digitally and integrates it into various information systems. One of these is the Genebank Information System. It is publicly accessible and enables anyone to search the Genebank's inventory online according to specific criteria. The classical Genebank at the IPK in Gartersleben is undergoing a process of change, from a pure collection to a Biodigital Resource Centre. As in the past, it not only preserves our crop diversity, but it now also provides comprehensive digital information, thereby making the genetic resources of our crops more usable and more targeted.